We're at the IAS meeting in Paris. We're here with Trevor Crowell, who's with the U.S. Military Research Program, and we just got done with a press conference, and I wanted to have you set us up on the work that you're doing, and then we'll discuss it a little bit.
Sure, absolutely. So RV397 was a randomized clinical trial of VRC01 monotherapy in a patient population that was treated during acute HIV infection. So it recruited participants at the Thai Red Cross AIDS Research Center in Bangkok, Thailand. A total of 18 participants entered the study and were randomized in a 3-to-1 ratio to receive either VRC01, which is a broadly neutralizing monoclonal antibody, or placebo. Those participants received their first study infusion on the day that they interrupted their antiretroviral therapy. So they had been well suppressed on antiretroviral therapy for at least 2 years. That therapy was started during the acute period of their HIV infection. And so they have small viral reservoirs, limited viral diversity, a number of reasons they'd be good candidates for testing of novel therapies to achieve HIV remission. They stopped that therapy, began infusions of study product, and we followed them for up to 24 weeks, during which they were serially receiving infusions, or if they experienced viral rebound or other reasons for ART resumption, then the study stopped and they re-began their antiretroviral therapy.
So where do you go next with this? This is kind of a start of the work that you're doing. So what is your next steps?
So broadly neutralizing monoclonal antibodies are being investigated for a variety of purposes. They're being investigated for use in HIV prevention, HIV treatment, and in strategies to achieve HIV remission. This was one...
You use remission, not cure, or is it similar?
They're similar, but I think in the case of HIV, the easier goal to achieve is remission in the sense...
Which is a period of time, not necessarily forever.
Exactly.
Yeah.
So we're not necessarily eradicating the infection from the body, but instead inducing a period of time when you wouldn't need the normal antiretroviral medications, when the virus would be controlled,
right,
not causing disease, but may still be in the body somewhere.
So an immunological effect.
Exactly. And that's what monoclonal antibody therapy is. It's an immune therapy. It's passive infusion of antibodies. And so this study was on that path, the HIV remission path, and I think it lays the groundwork for future studies that use not just one, but multiple monoclonal antibodies.
And then would you use another completely different approach to add to the effect, to maybe to make it more durable or long-lasting, or maybe prevention, total prevention?
Yeah, so I think that there are a few different strategies that need to be investigated. What's clear from the evidence so far is that one antibody is not going to be enough to induce remission or even to treat infection. So I think the first step is combining antibodies and using antibodies that target different parts of the virus.
So it's the virus, not the host?
Correct. The antibodies work against the virus.
Yeah.
So this particular antibody binds to the CD4 binding site on the virus and neutralizes the virus. And other antibodies work on other targets.
So you would have to have a specific type of virus for a specific type of antibody, or they're broadly?
Right, so they're broadly neutralizing, which means that in a laboratory, when you mix them with a variety of different viruses from many different people, they neutralize a large number, 90% or more. And so they target sites that are not unique to specific virus strains, so like the CD4 binding site, the V3 ligand, and others.
The sites are site-specific.
Exactly.
So it sounds exciting. I hope you can keep the work going. It looks like you've got a lot of work ahead of you.
Absolutely,
Yeah.
but it is exciting, and it's exciting to start to see even a small but some efficacy signal with these new therapies.
Right. So this is something that our U.S. government has been focusing on for some time, the NIH, and this is now, would you say, the next step? And then what are the next steps after that?
Yeah, so I mean, there are many people working on this, and much of the work is funded through the NIH and is done in collaboration with the NIH. The product that was tested in this study comes from the Vaccine Research Center, which is part of the NIH. And I think the next steps are looking at the combination therapies and also trying to enhance the antibodies that we have to make them more potent and longer lasting.
Is anybody actually accomplishing that, or is this kind of a hope?
It's very real. So the VRC which developed this antibody, VRC01, already has a next generation of antibodies in the pipelines that are more potent, more broadly neutralizing, and one key characteristic is that they're longer lasting in the system so that you don't have to dose them so frequently. And those are in preclinical studies now. It takes a while for a product to reach the point where it's safe to test in humans, but they're coming down the pipeline.
How do you make them more durable? Is this a sort of depot injection, or is it just that they just seem to last longer? They're in the body circulating and they just last longer.
Yeah, that's the latter is the case here, but certainly there are multiple ways to try and manipulate products to make them longer lasting.
Well, I look forward to more conversations with you. It seems like you're on the right track and early in the process, but nonetheless, I think the audience likes to know these are pieces of the hope that we have for maybe a cure down the line, but certainly for something that's, a treatment that's less needful of, constant pills and so forth, which, right now a lot of people are on. So
right,
a move to the next generation.
Yeah, there's a lot of people who are very interested in identifying therapies that are easier on patients, easier to take, and there's a lot of exciting work going on, and it's exciting to be a part of it.
And this is the future of that.
Yeah.
Great. Well, thank you very much.
Thank you very
it.
much.
Yeah.